Log on, tune in, find out. Another good idea from Cambridge.
Speaker in this session, who's Chris Brien from South Australia. He's going to talk about robust microarray experiments by design, a multiphase framework. Over to you, Chris.
Thank you. So, yes, it's back to design. This is the outline of my talk. I'm going to talk about phases in microarray technologies, then look at designing and analysing multi-phase microarray experiments, talking about avoiding bias and minimising variability, and then there'll be some conclusions. So just to make sure we all understand about what goes on in microarray experiments, I've got this little outline of the processes. So for something like a 2-channel spotted microarray, you start off with cells, perhaps one from a treatment and one from a control, or from 2 different treatments, and you extract the microarray from these cells, and then they are reverse transcribed and dyed so that you end up with some cDNA of different dyes. You have a chip which has got some probes on it that you perhaps make up yourself in the lab, and then you hybridize the 2 dyed samples onto the microarray, and then you scan it for colour. A similar sort of thing happens, although with differences, if you're using single channel like on oligo type arrays. In single channel, of course, you only start with the a single cell. You don't have the 2 treatments. You do extract RNA from those. You then reverse transcribe them to produce cDNA. There's usually another transcription process and a biotintylating, so that you end up with biotintylated cDNA, which is then fragmented. And again, you need to have an array, and these are often purchased, and have short oligonucleotides on them, and you hybridize the fragmented cRNA onto the chips that you have, and then you wash and stain them and scan them. So these are similar multi-step processes, I guess, with some differences, of course. So back at the last here in 2008, Terry Speed spoke at a workshop and talked about phases in multi-array experiments, and he challenged us to think about designs at every phase, because we don't usually think of them at every phase. So I decided that I would try and take up Terry's challenge, and so this is a first step in trying to do that. And I'm going to put forward a multi-phase framework that's based on a paper that myself and Rosemary and a couple of other colleagues have just about got published. It's available online. It's going into JABES, and it talks about when you've got a laboratory phase in an experiment, how you should design that. Well, you've got lots of laboratory phases in microarray experiments. In that paper, we define a phase to be the period of time during which a set of units are engaged in producing a particular outcome. And the outcome can be material for processing in the next phase, or values of a response variable, or both. And only the final phase must have need have a response variable. And in fact, in microarray experiments, that's usually true. That's the only phase that has a response variable. Everything else is producing something to go on to the next phase. And you can also have that one phase might overlap another phase. You don't have to have that they are separated discretely in time. Then multi-phase experiments consist of, obviously, several phases. Generally, multi-phase experiments randomise or randomly allocate the outcomes from one phase to the next phase. And for microarray experiments, while the need for randomisation is often mentioned in general terms, how to actually deploy it is not addressed specifically often in the literature. So the first thing is to think about some phases that you might have in a microarray experiment, and I'm going to give one list of phases and their outcomes that commonly occurs, but it's flexible. You don't have to stick with this set of phases. So I'm thinking here of a microarray experiment using the sort of process that I just described. It's going to start off with a phase in which you've got the selection or production of biological material, and what you produce is organisms or tissues that are going to be subject to the microarray experiment. Then you need to do the extraction, as we saw. So this is you need to get the samples and store them from the organisms or the tissues, and then you need to do the extraction, which will produce some extracts. Those extracts usually are labelled, so you'll end up with what I've called aliquots here from the labelling. You'll need some arrays, which you might either produce or purchase, and then you hybridise the aliquots to the arrays and perhaps post-wash the arrays, and then you would scan the arrays, the hybridised arrays, and that'll give you some measurements. So that's a list of each of these boxes here. Each of these represents a phase in the whole experiment. Now, when we talk about design in microarray experiments, we often think about design in this phase. That is, how do we assign the aliquots to the arrays to end up with the hybridised arrays? That's the main thing that we think about in microarray design. Well, actually, there might be also a design in the very first stage in the selection or production of biological material. It might be a field experiment, or it might be a animal experiment, or it might be even an observational study. And given that, it has been recognised, in particular Kathleen Kerr in her 2003 biometrics paper pointed out that microarray experiments could be two-phase, and as I said, the first phase can be an experiment or an observational, perhaps epidemiological study, and the second phase, this phase over here, is essentially a laboratory phase of the sort that we talk about in the paper I just mentioned. However, as Terry exhorted us, we should think about design in every phase. So in fact, there is potentially a design at every one of these phases, and so that's not usually considered. And if you do think about having a design in every phase, what you're going to do is end up with multiple randomisations, and that's something that Rosemary and I published about in a 2006 red paper, and you'd end up with multiple randomisations in a 7-phase process here. So we're really building the phases up. That's the most phases I've ever written down on one diagram. To sort of illustrate this process, I'm going to take an example, which is a sources of variability aphometrics microarray experiment. It was a study that was discussed in a paper by Zach Harkin and co-workers, and they were looking at the variability of labelling and hybridisation in microarray experiments. It involved 8 rats, and the phases are as follows. They had the production of biological material phase, and that produced a rat. They then took a sample, and the sample that they got was a mammary gland sample from each rat, so that was the product of the sample acquisition and storage phase. They then extracted RNA from that sample to end up with an extract. Then they took the extract and halved it for labelling. So each extract was halved. So in fact, when they came to do the labelling, they had 2 aliquots that they produced as a result of the labelling of the half extracts. Then they divided each aliquot again in halves for hybridising. So now they're going to end up with 4 aliquots half aliquots I should say, to hybridise. So they're going to need 4 aphometrics chips that they're going to purchase, and then they're going to assign I should say, the 4 half aliquots to the 4 hybridised arrays, and then they'll scan those and end up with measurements that will be measurements that relate to each individual rat. Okay, so that describes the experiment as they conducted it. They proposed a model like this. They had an observation was made up of a mean, or a mean, the grand mean, and then they had 3 random terms, one for rats, one for labellings within rats, and one for error. And you could write this symbolically in this way that I like to do. So we've got this straight line here. The fixed terms are on the left, which is just a grand mean, and we've got these 3 random terms in the model. And this model is equivalent to that for an ANOVA for a doubly nested design. We've got 2 hybridisations within 2 labellings within 8 rats. All very straightforward. Well, what about doing this in terms of the sort of multi-phase framework and thinking about the design of such an experiment? So the approach that I take is to first identify the sets of phases involved. That seems fundamental. consider how the outcome of one phase is to be assigned to the units in the next phase, which is not done necessarily. Then I'm going to produce a factor allocation description, which is something that we talk about in that paper, the Jabes paper. And then I'm going to formulate the full mixed model. I'll derive the ANOVA table and I'll use it to investigate the design and obtain a mixed model of convenience. So talking about designing these experiments, and I firstly want to concentrate on avoiding bias, essentially by randomisation, and I'm going to illustrate this, of course, using the example experiment. That's why I put it there. So we have the set of phases involved. I've identified those. These are the 7 phases. So how is the outcome of one phase to be assigned to the next phase? How am I going to assign the samples that I get to the extraction phase? What order am I going to do them in? Well, probably the simplest option I can think of if you're going to employ randomisation at every phase is just to simply randomise every phase. Okay, so given that we're doing that, what is the factor allocation description diagram for that? So what you need to do is take each of these phases and consider the factors that index the outcome of that phase, and it's pretty simple. So we'll have a panel for a set of objects, and here the objects are the set of outcomes. So as I said, we want to end up with a panel for each of the phases, and that panel will list the factors indexing the set. So we'll start off with the rat phase. The outcome of that phase are 8 rats, and it's pretty simple. We just need to index that with the factor here, which I have a capital letter for, with 8 rats. So an 8-level factor going from 1 to 8. So I distinguish between the outcomes here as being lowercase and the factor as having an initial uppercase. So all right, that's the first phase. Now in the next phase, I'm going to produce 8 samples, and I'm going to conduct 8 samplings to do that. And what I've said is that I'm going to completely randomise the order of the rats to the samplings. I'm not going to do I'm going to make sure they're in a completely random order. So in fact, what I do is do that, and I indicate that here by putting an arrow in that says that the 8 levels of the rats are randomised to the 8 samplings. Then we go on to the next phase, which is an extract, and I'm going to have to do 8 extractions. And what I am going to produce here is actually 16 half extracts, but I only need to consider the 8 extractions that I'm going to do at this stage. And what I will do is completely randomise the 8 samplings to the 8 extractions, because this is the simple design that I've decided to employ. So I do that, and then I divide each of my extractions into the 2 half extracts that it produces, and so I'll end up with a total of 16 half extracts. On to the labelling phase, I'm going to have to do 16 labellings, and this time I'm going to take the 2 half extracts from each of the 8 extractions, the combinations of those 2 factors, and label them to the 16 labellings, and then I divide the outcome of that into 2 half aliquots in each of the labellings, producing 32 half aliquots. I'm going to want to end up with 32 hybridised arrays, as I say over here, and I'm going to have to do 32 hybridisations. So I'm going to randomise the order of the aliquot halves to those 32 randomisations, another completely randomised design. And I'm also going to need some chips to be involved in each of the hybridisations, and what I'll do is get 32 chips and randomise those to the hybridisations as well, so that they're in no particular order. And then finally I've got to do 32 scans, and the order in which I do those scans, I'll completely randomise the 32 hybridisations to those 32 scans. Okay, so that's a factor allocation description diagram that describes the randomisation if you randomised everything at every phase. So I want to get a mixed model for that, and the way I do that is use the method that Clarissa Demetrio and I outlined in a 2009 paper in Jabes, which basically says from each panel you form the terms as all combinations of the factors subject to the nesting restrictions on the factors, that is what the factor is nested within. And for each term, you've got to decide whether to add it to the fixed or the random model. So I can start here with the first panel I've got up there, the rats panel. Well, I should say I'm going to have the grand mean as a fixed term, and then I go to the rats panel. There's only one term, that's rats, and I'm going to have that as random, so I put that to the right here of the straight line. Samplings, I've done the same thing. It's going to be random as well, so I add that to the previous rat terms. And then I'm going to move on to the half extracts panel here, and here I want to take all combinations of those two factors subject to the nesting, and there are only two terms, the extractions and then the extractions, what I say here, are wedge half extracts. So this is really stands for a 16 level factor, which is all combinations of the two half extracts with 8 extractions. I can't have half extracts on its own because it's nested within extracts. A similar thing for the labellings. I want all combinations of these two factors, and that of course given the nesting is labellings plus labellings wedge extraction, labellings wedge aliquot halves. And then I'm left with the 3 single factor panels here, and you'll have a term for each of those in the model. And that gives me the complete model for the experiment, taking into account all the sources of variation in this experiment. Now there is a bit of a problem because not all terms are estimable in this model, and you can use the ANOVA to show this, and so I'll do that. So what we do to produce the ANOVA is we actually start over here on the right and produce a column for each of these panels that has the terms related to the factors in the panel. So in fact for this first one here, scans, that's the only factor, so it's just going to be a term scans that has 31 degrees of freedom. It's almost a non-thing. There's no division of the variation. There's just this scans term. Then you'll go to hybridizations, and that'll be a next set of columns with source and degrees of freedom, which will be 32 hybridizations. So hybridizations with 31 degrees of freedom. But of course, these will be confounded, and I mean confounded here, not aliased, just to take up on Jeff Wu's earlier point. And so that you won't be able to separate those two terms, and similarly with chips you'll end up with a chips term with 31 degrees of freedom, and because I have only got a finite amount of room across here, instead of separating these into scans with 31, hybridizations with 31 and chips with 31, I've put them all together into one column here because they are all confounded in any case. But now we want to get the terms or sources in the analysis of variance coming from this half aliquot section, and we can see that the terms here, there will be 15 degrees of freedom for labellings, and for the aliquots within labellings there will be 16 degrees of freedom. And so we can add a set of columns that shows those two sources of variation. Of course those are confounded with the previous sources of variation, scans, hybridizations and chips. To the next panel, the half extracts panel, well, we're going to get a similar thing. We're going to have a term extractions with 7 degrees of freedom and half extracts within extractions with 8 degrees of freedom. But of course they're going to be confounded with labellings, and the way we do that is line this sort of sub-table I've got here up with the labellings to indicate that these two are confounded with the labellings. And then the next one is samplings, and samplings is just going to be a single term with 7 degrees of freedom but confounded with extractions, so I line that up with extractions. And then finally I have the rats, and of course that will be 7 degrees of freedom lined up with samplings. So that's my analysis of variance table, and the thing about this is that you can show that what happens here is you've got rats and samplings and extractions that are not separable. So in fact you can only measure the sum of those, and they do represent the biological variation. And similarly you can't separate half extracts from the labellings, so you can only estimate the sum of those, and you can't separate the aliquot halves from the chips, hybridizations or scans, so you can only estimate the sum of those. So in fact there's really only 3 things you can estimate here, and to be able to get an analysis you need to select one from each of these groups to give you a model that will actually run in mixed model software. This last term here is referred to as error by Zach Harkin, but you can see here, we can see that what this error is made up of. It's made up of variation from aliquot halves, from chips, from hybridizations and scans. So the aim here is a model in which all the potential sources of variability are identified, not one in which all are separately estimable. And the mixed model for convenience, well, as I say, I selected one from each of those groups, so I end up with rats extractions, half extracts combined with extractions, and aliquot halves labellings. And that is equivalent to the Zach Harkin model. The second aspect is minimising the variability, and we know we want to do that so that the variance of the treatment estimates is as small as possible. And as usual, there are three possibilities for this. You could have stringent experimental protocols to reduce the variability. You can have experimental design to avoid the variability, and statistical analysis to adjust for the variability. The stringent experimental protocols, well, several authors have investigated variability in different phases, including Zach Harkin, but there are others, and they found some phases, in particular the hybridisation phase, is more variable than others. So you see that people like Han and his co-workers advocate minimising the variability in the hybridisation phase as a way of overcoming this sort of variability. So they put in place more stringent protocols. From an experimental design point of view, you really need to take into account batches is often stressed in the literature, and Altman in a paper in 2009 lists sources of batch effects in all potential batch effects in every phase. So it's quite possible, does happen quite a lot. And indeed the big MAQC study in their standard operating procedures stipulated that if you can't do all the processing in one day, please batch it, and they had recommendations of how to do that. So batching is definitely something that is seen as important. In fact, there's a book on that Naomi Altman paper is out of a book on, it's called about batching in microarray experiments. And generally one looks for batches built into the process, so if there are different acquisition times of samples, or there's different processing days, or different operators, or different batches of reagents, or sets of simultaneously processed specimens, they're all obviously going to lead to batches. And of course, if you've got treatments, you want to block them according to batches. So I just thought I'd look at this with that example and think about batching in the example, not that it was thought about in the paper. And let's suppose for each rat the tissue be obtained and the RNA extracted immediately. That is, we're not going to randomise the order of RNA extraction, we're going to do it in the order in which we sample the tissue. In the labelling phase, let's suppose 1/2 extract from all the extracts will be labelled in one batch and the remaining ones in a second batch. I don't think they did this, but it would actually be a good thing to do because you'll get a better estimate of the variability arising from the labelling process, because if you do them both together, it's likely to be more similar if you stretch them out. So you're likely to get a more realistic estimate of the variability from labelling. And I'm just going to suppose that this batching went through into the hybridisation phase, that is the batching and the labelling phase, where washing, say, occurs in batches of 16. And for practical reasons, suppose that the 2/2 aliquots from an aliquot are hybridised consecutively. In the scanning phase, I'm just going to suppose they all happen in a random order. So we're going to start off with our 8 rats, and then we have our 8 samplings, and I'm still going to randomise the order in which I sample the rats. But then I'm going to extract RNA from each of the samples, but I'm not going to do that in a random order. I'm going to do it in the order in which I do the sampling. And so to indicate that, I put a dashed arrow in that indicates this is a systematic factor. It's done in a systematic order. It follows, in this case, the order in which the samplings were extracted. And then I divide those the results of those extractions into half extracts, and it gives me my 16 half extracts. Now in the next phase, what I said I wanted to do was I wanted to take one of the extracts from each of the 8 extractions and do those all together, and then do the other half of the extracts. Now there's a bit of a problem here because actually I've got 2 half extracts from each of the 8 extractions. That's 16 half extracts that I somehow want to apply over here to the two batches, and you can't sort of do it in that way. The way to get around this problem is to introduce pseudo factors. So what I'm going to do is introduce a pseudo factor that I've called H1, H for half extracts, so it labels the half extracts and is a pseudo factor of half extracts. And it's a two-level factor, and what we do is we give one of the half extracts from each of the extracts we assign level 1, ideally at random, and then the other half extract from an extraction gets level 2. And so I've now got them labelled level 1 and level 2 according to H1, which is then randomised to the two batches, and then I can randomise the 8 extractions in each level of H1 to the 8 labellings. And we then divide the 2 aliquot halves in each labelling in each batch into 2. I then want to do I want to produce my 32 hybridised arrays, and I said I'd follow the same sort of batching as I had in the 32 aliquots, and to do that I need to divide the production of those arrays into 2 occasions during which I have 8 periods during which I do 2 hybridisations. And then I can assign the 2 batches to the 2 occasions at random, and then I can assign the 8 labellings to the 8 periods at random, and I can assign the 2 aliquot halves to the 2 hybridisations in each period at random, and you can see that what's going to happen is that you're going to get the 2 aliquot halves done consecutively, as I said. You're going to need 32 chips that have to be have the aliquots from the extract. What do I call them? I can't remember. I need to randomise these 32 chips to the combinations of those factors so that there's no particular order there as well. And the way I show this is that I am going to take the 32 chips and randomise them to all combinations of the factors. That's what this bullet with the 3 arrows leading from it signifies. And then I want to do the scans in random order, so I have to randomise the combinations of the 3 factors to the 32 scans. Mixed model, grand mean, the same terms as I had before because nothing has changed here essentially about the factors involved, but now I need to consider what are going to be the terms in the model here, and it's the same sort of process as before, and we end up with these 3 terms which are the combinations that respect the nesting in the design. And similarly in the hybridised array panel, I have the 3 terms, occasions, occasions periods, and occasion periods and hybridisations. And then we add in the terms for chips and scans as well, and that gives me the full model. Of course, that's not going to fit, but actually this one will. And the ANOVA is a good thing for showing that, but I obviously don't have time to go through the ANOVA that would apply in this case. So the conclusions are, is it a good thing to do? Well, it's a lot of work. Maybe I randomise the rats in the start. Isn't that enough? It's a lot of needless redundancy, perhaps, such a lot of factors that you don't sort of need in a way. But the pros are, encourages consideration of the appropriate design in all the phases, even if ultimately it's decided not to randomise all the phases. As for all experiments, in a microarray experiment, randomising it makes it robust to systematic biases in that phase. Often processing orders not considered, perhaps assuming no systematic change during a phase or processing batch, but I sort of wonder if it's tenable. If you're always doing the material from the same rat at the start of every phase, perhaps that's during operator or equipment warm up and randomisations and ensure against effects like that. Blocking based on batches obviously assists in minimising variability, and I think this sort of approach promotes the identification of all the sources of variability at play in a microarray experiment, even if not all are estimable. And this is a summary of what I've just talked about, and there's some references here to the paper, and it'll end up on the multi-tiers site. Thank you.
Thank you very much for that, Chris. We have time for one question or comment. Dennis.
Keep hearing the keyword randomize. Could you say a few words about what did you do?
Sorry?
Could you say exactly how did you do for randomize? When you say
Well, I'm going to do a randomisation at every phase that I decide to do a.
Precisely what do you mean by?
Well, I would.
Point you, I don't know. I mean,
Well, for example, I don't know if I can do this quick. You know, if this, what I would do here is I would have a list of rats from 1 to 8, and then I would assign the sampling values to that in a totally random order. And then at this phase, I would then I'd put extractions, and I'd have to assign that in a completely random order to samplings.
well, I'll give you background about the question because 2 weeks ago we have a small group discussed about this, and there's some question about the quality of randomization. That's the issue I'm trying to raise here. But nonetheless, it could be.
Oh, well, I'd be interested to talk to you about that afterwards, perhaps.
Yeah, I think, again, there'll be plenty of time to discuss this more within the wine this evening or at other times during the week. So we need to move on to our last talk. So thanks once again, Chris, for that.